Hey everyone, in this video we're going to be talking about factors that influence the rate of an enzymatic reaction. Those include temperature and pH, and then we're going to look at inhibitors of enzymes. And there are three types that we'll look at: competitive, non-competitive, and irreversible. And by the end of the video, you should be able to understand how temperature and pH affect an enzyme reaction rate, and you should understand the different types of enzyme inhibitors. So first, let's look at temperature and enzyme activity. So enzymes have a optimum temperature at which they prefer. And this varies from organism to organism, depending on the body temperature of that organism. Humans have a body temperature of 37 degrees Celsius. And so all the enzymes in our body have evolved to operate best at 37 degrees Celsius. Organisms that live in the Arctic where it's freezing cold all the time, their enzymes, are used to it freezing cold, and that's like their ideal. Some extremophiles live in like hot geyser water, around thermal vents, and they've evolved to where their enzymes operate best close to 100 degrees because they, live in boiling hot water. So it varies from organism to organism, but for any given enzyme, there is an ideal temperature that that enzyme prefers. For human enzymes, it's 37 degrees. Now, if you decrease the temperature, the enzymatic activity will decrease. That's because enzymes move slower when it's cold. So an enzyme has to be flexible, like we saw for the induced fit model. Well, if you freeze an enzyme, it will flex slower, which means that the turnovers become slower. And so you just get a slower overall reaction when the enzyme is cold. Now, if you heat the enzyme up, you get a drastic decrease in activity. And that's because over 50 degrees Celsius, the enzyme protein will start denaturing and unfolding, and then you've got a dead enzyme. If it denatures, it no longer has the same active site. It can't catalyze the reaction anymore. It's useless. It's a dead, denatured enzyme. So cold temperature slows it down. Hot temperature denatures and kills it. Now, pH is kind of similar. There's a Goldilocks zone, right? The enzyme prefers an optimal pH. Anything too far below or too far above denatures the enzyme, and so you get no activity. Most enzymes in the human body have a optimal pH around 7.4, because that most of our internal body is about 7.4, but it can vary depending on the specific tissue. And so here's a chart showing you different enzymes and where they're located in the body and what their optimal pH is. So a really obvious one is pepsin, which is a enzyme that breaks down protein in your stomach and small intestine. So when you eat a steak, there's an enzyme in your stomach that starts breaking down the proteins in the steak. That's pepsin. Well, you have stomach acid, and so pepsin has evolved to operate best in an acidic environment because it lives in your stomach. And so his ideal pH is like 1.5 to 2. But that's a unique example. Most enzymes are around 7. Another unique one is arginase, which lives in the liver. Your liver has certain regions that have a very high pH. They're really basic. And arginase exists in those tissue types, and so it prefers a pH of 9.7. So ideal pH is unique depending on the tissue type, where that enzyme is located. But for the most part, human enzymes prefer about 7.4, which should have been what we noticed in lab this week. Your enzyme reaction, when you did it at the different pHs, should have worked at pH 7. And then when you got too low or too high, you had no reactivity at all because you were denaturing the enzyme. So the next thing we're going to talk about are inhibitors. So an inhibitor is something that stops an enzyme from functioning. The way they work is they stop an enzyme from binding to its substrate. So normally an enzyme and a substrate form an enzyme substrate complex, and then, it can convert the substrate into product. An inhibitor will bind to the enzyme and form an enzyme inhibitor complex. Once the inhibitor is bound, no product can get formed. So let's take a look at competitive inhibition. Competitive inhibitors have a structure, like a 3D shape, that is similar to that of the substrate. And because they have a similar shape to the substrate, they bind to the active site of an enzyme. Because they bind at the active site, they are in competition with the substrate to see who's going to get in the active site first. It's kind of like musical chairs, right? You're both trying to sit on the same chair. So you're competing with one another. So normally the substrate binds in the active site and everybody's happy. But if the competitive inhibitor gets in the active site first, then the substrate can't get in there and you don't make any product. Now, this is the least severe type of inhibition. And the reason is, if you just had more substrate molecules, the statistical probability would be that the substrate would bind the active site. So once again, thinking about musical chairs, if you're playing musical chairs with teams, and you have 50 people on team substrate and one person on team inhibition, the sheer probability means that when the music stops, since there's 50 of team substrate, one of them is going to get the active site because they outnumber the inhibitor. And so the severity of the inhibition just depends on the relative concentrations. If you have a lot of inhibitor molecules floating around, you're probably going to get it binding. If you have a lot of substrate molecules floating around, then you're probably going to get substrate binding. And so competitive inhibitors are often like mild poisons, like in foods, where you might get sick if you eat the food, but you're only sick for a little while and it kind of goes away. It's only really serious if you eat like a ton of it. And that's because it's competing with a regular substrate. And so it's only really dangerous if you have a lot of that competitive inhibitor in your system at once. Now, the second type of inhibitor is called a non-competitive inhibitor. Non-competitive because it's not competing for the active site. Non-competitive inhibitors bind on the outside of the enzyme, not at the active site. And so this is like playing musical chairs with a cheater. They flip the chair over and sit on the other side of it. So there's no more chair for the substrate to sit on. These are a little more serious, right? So even if you have 50 substrate molecules floating around, that one non-competitive inhibitor can still, flip the musical chair over and then nobody can sit on it, even though there's more of them on that team. And so these are like poisons that are very potent, okay? Like the food that the mushroom, you eat one of the poisonous mushrooms and you could die from it, even if you just ate one. It's because it doesn't matter how much substrate you have, that non-competitive inhibitor binds on the outside of the enzyme and there's nobody trying to stop it. And the reason why it blocks the active site, even though it's not binding the active site, has to do with conformational changes of the enzyme. When the non-competitive inhibitor binds on the outside, the entire enzyme induces a different fit to try and fit the inhibitor. And when it induces that fit in the other direction, the active site opens up and it's too open, and now the substrate can't bind anymore. So non-competitive inhibitor binds on the outside, changes the shape of the enzyme enough to where the substrate can no longer bind. These are more serious because it doesn't matter how much substrate you have. they're not competing for the same spot, so it will always be able to bind on the outside. So more dangerous. Now, the last type of inhibitor is called an irreversible inhibitor, and these are the most dangerous. These are like those super lethal poisons, like you get a little bit of it in your system and there's no cure. You're just going to die. There's certain poisonous mushrooms where like you eat half of the mushroom and you'll die 2 days later, even if you go to the doctor. Once it's in your system, it's permanently binding to certain enzymes. It takes a couple days for you to die, but like there's no cure because it permanently, irreversibly blocks certain enzymes. And so the reason why they're permanent is they form a covalent bond to the enzyme. So most of the time when a substrate or a competitive or non-competitive inhibitor binds, it's not permanent. They fit because of intermolecular forces, but they can pop back out. Irreversible inhibitors form a covalent permanent bond. And so once they bind to the enzyme, they're never going away. And that whole enzyme is destroyed. It's done. The only way to recover from this is for your body to make completely new enzymes, which you can do if it's not, that serious. But if you get a hefty enough dose and every single one of your enzymes gets permanently knocked out, that could be lethal. These tend to be found in like insecticides or antibiotics or nerve gases, things that are extremely lethal to the organism that they, attack. The most common example that textbooks give is penicillin. So penicillin is an antibiotic. It's really deadly to bacteria, but it doesn't affect humans. And it's an irreversible inhibitor of the enzyme that makes the bacterial cell wall. So it forms this ester bond here to the active site of the enzyme, and then the enzyme can't, pick up its substrate anymore. Now, humans don't have cell walls the same way bacteria do, and so we don't even have that enzyme in our body. So for us, penicillin is, does nothing to us, but to a bacterium, it's deadly and it's permanent. They can't undo it. So just an example of a type of irreversible inhibitor. There are many, many types of irreversible inhibitors. And so the last thing I'm going to show you here is this summary slide. And it shows you the 3 types of inhibitors and their characteristics. So the shape of an inhibitor. A competitive inhibitor is the same shape as the substrate because they both bind the active site. Non-competitive and irreversible have totally different structures. The location where they bind to the enzyme. Competitive binds at the active site, while it's competing. Non-competitive does not bind at the active site. And an irreversible can bind anywhere. It could bind in the active site or it could bind on the outside of the enzyme. The key feature is that it forms a covalent bond and it's permanent. And so the last topic is reversibility. So competitive inhibition can be reversed if you just have more substrate floating around. Non-competitive is not reversed by having more substrate because it's not competing. But eventually you can remove it from the enzyme. It's not a permanent thing. And then irreversible, they're permanent. You can't undo it. You just have to make new enzymes in your body. That's it for this short video. And this is the last video for the semester. So I appreciate you guys watching along with me throughout the semester. And I will see you either in office hours or next week for the final exam. Have a good one.